Welcome back to the third video of episode 2. We're going to cover the mechanism of inspiration and expiration, the use of a bell jar to try and model exactly how that works, and we'll include the structure of the alveoli, which you need to know. You need to be able to label that diagram to explain what makes it really good at its job, and then be looking very briefly at how the composition of inspired and expired air differs. You can probably already guess how that differs. I think we've mentioned it before, in fact. So here we have the respiratory system, which we've seen before. Remember, you need to be able to label each of these, the labels indicated here. Okay, so we're talking now about inspiration and expiration, exactly what goes on. So this is the main muscle of inspiration, the diaphragm. Check that fairly unusual spelling there. And what actually happens is when you breathe in, this muscle, this sort of parachute-shaped muscle, which is several millimeters thick, when this contracts, it actually flattens out and pushes downwards. So it would change more towards this sort of shape, and that pushes on the abdominal contents. And along with these muscles, the intercostal muscles which contract, these will pull the ribs up and out. Not a great deal, but when they're all involved, what actually happens here, we have a huge increase in the volume of the chest cavity. This now from the side. So here's inspiration. There's the diaphragm, and you can see that arrow there is indicating that the diaphragm contracts and pushes downwards. The intercostal muscles, they will also contract, so the thoracic cavity, the chest cavity, will expand. Okay, volume has increased. So if the volume has gone up, okay, that has increased, then the pressure has decreased. So there is less pressure inside the lungs than there was previously. Now, the pressure of the atmosphere here is going to remain the same. This pressure is changing. So if the pressure inside the lungs becomes less than atmospheric pressure, then air is going to rush in to those lungs and fill those lungs up, which is inspiration. Okay, now expiration, all because of the elastic properties of the thoracic cavity and of the muscles involved, all we do is relax at rest. The diaphragm will go back to its previous shape. The ribs will move down and in, so the volume will decrease, which increases the pressure. So pressure becomes more inside the lungs than in the atmosphere, so air is therefore going to rush out in that way. To really help us understand this, we'll use this sort of setup in the classroom, certainly. So what we've got is a bell-shaped glass jar, simply obviously called just a bell jar. We've got it sealed with a bung, and we've got just a tube coming down that splits in two. Okay, and then over the tubes you've got the balloons, and the idea is you want a small amount of air inside them, and then you sort of seal the jar with a rubber diaphragm, okay, which is normally just a balloon or something itself. The idea is you grab hold of that rubber diaphragm, that balloon, and you pull it downwards. As you pull it downwards, the volume inside this area is going to increase overall, therefore the pressure will decrease. So if the pressure goes down, the pressure is decreasing, then the pressure inside this area is less than in our atmosphere, in our environment, so air is going to rush in through that tube, through there and the other way, and into these balloons, and therefore these balloons will slightly inflate. And so this is a wonderful model to try and explain how it is that the lungs can fill up in the way that they do as a result of the movement of the diaphragm. Okay, so we pull the diaphragm down, volume increases, pressure decreases, therefore the air will rush into the tubes and fill up our balloons in that way. Now if I was to release the diaphragm, so I stop pulling that downwards, it will return to its previous shape, which in reality would probably be straight across, which will decrease the volume, increase the pressure, and therefore air will rush back out the way it came. And what I would normally do is pass that around the classroom, give everybody a chance to have a look at that. Okay, so this tube there represents the trachea, we've got the bronchi, we've got the lungs. The bell jar itself is to represent the chest wall or the rib cage, and the rubber diaphragm, the rubber balloon, represents the diaphragm muscle. Okay, so this is a very, very good model for explaining how the lungs can inflate and deflate as a result of these movements. You could be asked to evaluate this as a model. So if it asks you to do that, what you're going to try and do is make a few points about why this is a good model and a few points about why this is a not so good model. Okay, so the good points are we're representing these structures: trachea, bronchi, lungs, rib cage. You pull the diaphragm down and it helps us explain the changes that take place in volume and pressure. So you could list those good points. The bad points, well, this rib cage, if it was a rib cage, isn't moving. The airspace within there as such doesn't really exist in the real model, in the real situation. Okay, and we don't have any intercostal muscles, and we don't have any bronchioles or any alveoli represented at all. So you could kind of pick holes in that. Although it is a very good model, obviously it is limited in just how accurate it is compared to the real life situation. Now, if we were going to predict what effect we think this might have on the oxygen and carbon dioxide levels in expired air compared with inspired air, just think back to your equation of respiration. So we've got glucose plus oxygen. So we need oxygen, aerobic respiration, and that's going to be producing CO2 plus our water. So it's going to be taken in, it's taken out of the inspired air oxygen, and in expiration it's going to be putting in carbon dioxide. So hopefully you're already making a prediction that the oxygen levels are going to be less in expired air and the carbon dioxide levels are going to be higher in expired air. And that is exactly the case. Okay, so if we breathe in 21% oxygen, we would normally breathe out 16%. Okay, you might see quoted in some textbooks, they do quote 17% there as a result of rounding up, or depending on the way they've measured things. With the carbon dioxide levels, we inspire 0.04% and we expire, we breathe out 4%. And again, depending on the textbook you look in, you might see 0.3% and 3% quoted there, which I have seen previously. Okay, now nitrogen is 79% breathed in and the same breathed out, because we make no use of nitrogen in this form whatsoever. We can't touch that nitrogen, we just breathe it in and breathe it out unchanged. The water vapor content will vary depending on your environment. Okay, so that could be depending on the humidity of the air that you're breathing in, but you will breathe out saturated air, which you'll notice on a cold day, because when you breathe out, that water vapor will condense and you'll see that as the mist as you breathe that out. Okay, now the structure responsible for all these changes is the alveoli, and you can see that structure in the diagram before you now. We're looking at, if we follow the path of the air in through the mouth, through the nasal cavity, we're going to come down through the trachea into the bronchi, into bronchioles, which will further split in two, and eventually you will get to the end of the bronchiole and you have these structures here which are called alveoli, or one alveolus. Okay, so as you can see, these structures are very well suited to gas exchange. They have a thin film of moisture because the gases will diffuse much more quickly when dissolved than they would in dry air. The very, very thin wall, that's just one cell thick, as is the capillary. So the oxygen, which is going to be in a high concentration inside the alveoli, can diffuse very easily into the blood in that way, and then into the red blood cells. So we've got deoxygenated blood which is lacking in oxygen or low oxygen levels, And as it flows this way past the alveoli, it's going to be picking up oxygen and become oxygenated. Okay, so a thin film of moisture, we've got a very thin wall, we have a very, very good blood supply. And the other thing that makes alveoli so very good at their job is the fact that there's just so many of them, that the surface area for them to actually diffuse oxygen into the blood is huge. Okay, so if you add up the very many millions of alveoli in your lungs, there's a huge area for them to diffuse in. Okay, and remembering, of course, we've also got carbon dioxide diffusing out. Whoops. You could see that coming in through there. So once again, this is yet another diagram that you'll have to be prepared to label, and as I've said many times before, the diagram you get is unlikely to look anything like I've shown here or any textbook you look at. It could be one you've seen before, but it's more likely to be one unique to the exam. So you need to be able to recognize each of the things that we've mentioned there and the features that make the alveoli so very good at absorbing gases and allowing diffusion of gases, oxygen into the blood, carbon dioxide out. I just want to finish with a mention of the effects of smoking. So there are three main things we need to look at. Primarily, smoking immediately its impact on cilia and mucus. Okay, something called emphysema. And lung cancer. Back to the lining of the trachea and the bronchi and bronchioles, we have these specialized epithelial cells with these tiny structures called cilia, these hair-like structures, which constantly push up the mucus to the top of the throat, where any particulates, any microbes that have been trapped are actually then swallowed down into the stomach. Now, if you smoke, one of the effects is to actually stop the cilia from moving. So that process of pushing the mucus up to the top of the throat will stop up to something like 40 minutes. So if somebody was to have a cigarette every 40 minutes during the day, which I don't think is that unusual for some people, then their cilia would go all day without cleaning the lungs, without pushing these particulates up and out. And that gives these, if there are microbes, if there's bacteria in here trying to grow, that gives them a really great opportunity to get a foothold and start growing. Another effect will be to actually dry out the lungs and stop the production of mucus, which, as you can imagine, because this is the stuff that's actually sticky and causes those particulates, those microbes to be trapped, without the mucus, even if the cilia are working, you can see their job's going to be much, much harder, and it's less likely to clean out the lungs in those ways. So the first effect to talk about with smoking is the fact that the lungs will not get cleaned, and then you're much more likely to pick up infections and to be coughing. And when you do cough, it's going to be very violent, because obviously you're trying to clear out a day's worth of filtered microbes, bacteria and particulate and all the bits and pieces we've breathed in throughout the day. Now with the repeated coughing, okay, so if I've tried to draw on here the normal alveoli, always kind of reminds me of a bunch of grapes, I guess, and that's what I've tried to draw there. So we've got lots and lots and lots of these individual alveoli being shown up there, which gives a huge surface area for gas exchange to take place. But if you're constantly coughing and coughing very violently for many weeks and months and years, what actually happens is that these alveoli start to flatten out. And because they flatten out, that really reduces their surface area. So if there's a less surface area for gas exchange, that means you're less good at getting oxygen into your blood and less good at getting rid of carbon dioxide. And that has all sorts of effects on the body, anywhere from parts of the body being starved of oxygen to a buildup of carbon dioxide, which can affect the pH levels in your blood, which can have all sorts of knock-on effects to your bicarbonate levels and all sorts there as well. Okay. And just to mention finally, I've no diagram to show this really, But lung cancer, there are many chemicals in smoke which are carcinogens. That means they cause cancer. Okay, so carcinogens. So they cause cells to become cancerous and increase your chances of that. And looking on the NHS website, I think it was 84%, but in the WJEC specification it does quote 90% of cases of lung cancer occur in people who smoke. Okay, so if you're asked what are the effects of smoking and why shouldn't you smoke, then I would start by talking about the cilia and how these don't move around and they don't clear out the lungs and the effects of that. The possibility of developing emphysema from the lots of the smoker's cough, as I guess it would be called, and finally the vastly increased chances of getting and developing lung cancer as a result of smoking.